6 Craziest Cases of Mind Control in Nature. Here are 6 of the craziest and most fascinating examples of mind control in nature, showcasing the incredible adaptations of parasites and symbiotic organisms. Hairworms. Hairworms, also known as nematomorpha or Gordian worms, are fascinating parasites known for their ability to manipulate the behavior of their hosts in extraordinary ways. Hairworms typically infest terrestrial insects such as crickets, grasshoppers, and beetles. They lay eggs in water, hatch and are consumed by aquatic organisms, which are later eaten by terrestrial insects. Inside the insect, the larvae develop into adult worms by feeding on the host's internal nutrients. Hairworms need water to reproduce, but their insect hosts are typically terrestrial and avoid water. Once the worm matures inside the host, it manipulates the host to seek out water and jump in. Hairworms secrete molecules that affect the host's central nervous system. They may alter neurotransmitters like serotonin or dopamine, disrupting the host's natural instincts. This causes the host to behave unusually, such as actively moving toward water. Once the host enters water, the hairworm emerges, often killing the host in the process. The worm then reproduces, completing its life cycle. The ability of hairworms to control their host's behavior has made them a topic of interest in parasitology and neuroscience. This type of parasitic control highlights the complex interactions in ecosystems and raises questions about the evolutionary advantages of such mechanisms. It also provides insights into how chemical signals can drastically alter behavior, with potential applications in medicine and pest control. Gpoppantal WASP. The Gpoppantal WASP, a parasitoid wasp, is another fascinating example of a mind-controlling parasite in nature. This species manipulates its host, typically caterpillars, to act in ways that benefit the wasp's offspring, often at great cost to the host. The female Gpoppantal wasp lays dozens of eggs inside a caterpillar, such as a moth larva. The eggs are deposited directly into the caterpillar's body, and the larvae hatch and begin feeding on the host's internal organs. Remarkably, the larvae avoid vital organs, keeping the host alive. The wasp larvae grow inside the caterpillar, feeding on its internal tissues. Once the larvae are mature, they chew through the host's skin and emerge to form cocoons nearby, leaving the caterpillar alive but significantly weakened. The precise mechanism by which Gpoppantal wasps manipulate their hosts is still under study, but researchers have uncovered some clues. The larvae may release chemicals that alter the caterpillar's nervous system, effectively reprogramming its behavior. These secretions could interfere with neurotransmitters, similar to how other parasites manipulate their hosts. It is hypothesized that some larvae remain inside the caterpillar, influencing its behavior until the cocoons are ready to hatch. This behavior ensures the survival of its vulnerable pupae, which are protected by the host's efforts. There is no direct benefit, and the manipulation ultimately leads to its death, highlighting the wasp's exploitation of the host. The Gpoppantal wasp provides a fascinating case study in parasitism and behavioral manipulation. Euhoplorchus californiensis. Euhoplorchus californiensis is a fascinating parasitic flatworm known for its ability to manipulate the behavior of its host, the California killifish, in ways that increase its chances of completing its life cycle. The life cycle begins with eggs released into the water, typically through the feces of birds that serve as the parasite's definitive host. The eggs hatch into free-swimming larvae called myricidia, which infect their first intermediate host, snails. Inside the snail, myricidia develop into sporocysts, which reproduce asexually to produce cercariae. Cercariae are then released into the water, where they seek their second intermediate host, the California killifish. Cercariae penetrate the skin of the killifish and migrate to its brain, particularly attaching to the brain's meninges, the protective layers surrounding the brain. Here, the parasite forms a cyst-like structure called a metacercaria, which can remain in the fish for an extended period. The killifish, under the parasite's influence, exhibits unusual behaviors that make it more likely to be predated by birds, such as swimming closer to the surface and performing conspicuous jerking movements. When a bird eats the infected fish, the parasite completes its life cycle, matures into an adult worm, and lays eggs that are excreted in the bird's feces. This behavioral manipulation significantly increases the likelihood of the parasite being consumed by its definitive bird host, ensuring reproductive success. Acacia trees. Acacia trees don't directly engage in mind control in the way parasitic organisms like hairworms or wasps do, but they employ fascinating biochemical strategies to influence the behavior of animals and even other plants in their environment. Acacia trees use chemicals and symbiotic relationships to deter herbivores and, in some cases, manipulate animal behavior to their advantage. Acacia trees produce tannins and other bitter compounds in their leaves when grazed upon. These chemicals make the leaves less palatable or even toxic to herbivores. For example, animals like giraffes often move on to other trees after feeding on an acacia, minimizing damage to any single tree. When an acacia tree is attacked by herbivores, it can release volatile organic compounds into the air. Nearby acacias sense these chemicals and ramp up their own tannin production preemptively, creating a network of chemically defended trees. Some species of acacia trees form mutualistic relationships with ants, which can resemble a form of mind control. The tree provides ants with shelter and food. The ants, in turn, aggressively defend the tree from herbivores and even prune competing vegetation. The tree's chemical signals ensure that ants stay dependent on the tree. By tailoring the nectar with specific nutrients or alkaloids, acacias can limit the ants' ability to forage elsewhere, keeping them loyal protectors. While acacia trees don't control the minds of other organisms, their ability to manipulate behavior through biochemical and ecological strategies has parallels to parasitic mind control. They modify herbivore and pollinator behavior. They influence ant loyalty to serve their survival needs. Phorid flies. Phorid flies, particularly those in the genus Pseudactian, are notorious for their gruesome and fascinating parasitic behavior, which can resemble a form of mind control. These flies, often called ant-decapitating flies, manipulate the behavior of their host ants in ways that benefit their own reproductive success. Here's how they work. Female phorid flies lay their eggs directly into the bodies of ants, typically in the thorax or head. They often target fire ants, Solenopsis species, a behavior that has made them a tool in biological control against invasive fire ant populations. After hatching, the larva migrates to the ant's head, where it feeds on the muscles and tissues that control the mandibles. The larva avoids vital organs initially, keeping the ant alive while it continues to grow. Once the larva is ready to pupate, it appears to manipulate the ant's behavior. The infected ant often leaves its colony, isolating itself from the group, which reduces the risk of the colony detecting and removing it. The ant may wander aimlessly or exhibit uncoordinated movements, a stark deviation from its typical behavior. This isolation benefits the fly larva, providing a safer environment to complete its development. The larva secretes enzymes that weaken the connection between the ant's head and thorax, effectively causing decapitation. The larva then pupates inside the decapitated head, eventually emerging as an adult fly. The exact mechanisms by which phorid flies manipulate ant behavior are still under study, but several hypotheses exist. The larva might secrete chemicals that interfere with the ant's nervous system, causing disorientation and unusual behavior. The feeding larva may damage key neural tissues, leading to altered locomotion and social behavior. Some parasitic behaviors may stem from an evolutionary adaptation where the larva triggers specific ant behaviors to maximize its survival. CulinaCulina. CulinaCulina, a genus of parasitic barnacles, is one of the most extreme examples of biological manipulation in nature. These parasites infect crabs and hijack their bodies and behaviors, essentially turning them into zombie servants dedicated to the parasite's reproduction. Here's how CulinaCulina exerts its incredible mind control. The life cycle starts with a free-swimming larval stage called a cypress larva, which searches for a host crab. Upon finding a crab, the female larva attaches to a joint in the crab's exoskeleton and injects cells into the crab's body. These cells develop into a root-like structure, called the interna, that spreads throughout the crab's body, taking control of its internal systems. The interna grows and consumes the crab's nutrients, effectively taking over its body. Meanwhile, a reproductive sac, the externa, forms outside the crab's body, often near its abdomen, where the parasite releases its eggs. The parasite chemically castrates the crab, preventing it from reproducing. This ensures that all the crab's energy is redirected to supporting the parasite. CulinaCulina manipulates the crab's behavior to treat the parasite's eggs as if they were its own. The infected crab grooms and protects the externa. When the parasite releases its larvae, the crab performs behaviors similar to releasing its own offspring, such as waving its claws to help disperse the larvae. Culina infects both male and female crabs, but it feminizes males to make them behave like females. The parasite alters the male crab's hormonal balance, suppressing masculine traits like large claws or aggression. It induces the male crab to develop a wider abdomen, resembling a female's brood pouch, to better carry the parasite's eggs. This manipulation ensures that male crabs can still perform the maternal behaviors necessary for the parasite's reproductive success. By altering crab behavior and population size, Culina indirectly affects the broader marine ecosystem, including predator-prey relationships and habitat structure.